Hi, micro folks. This is a follow-up for Lab Manual Chapter 10. These are our blood auger results. And again, my setup here, folks, it's going to make it really hard to see the hemolysis, but what I'll do, I'll just show you the plates and then we'll do a follow-up PowerPoint which will have some nice images. So folks, you'll recall that we said that blood auger, it's enrichment so that we can grow fastidious bacterial pathogens, supplying all their growth factors. So usually in diagnostic labs, a sample coming in from a human patient, if a bacterial pathogen is suspected, you're always going to streak to or inoculate blood auger. So folks, this is a blood auger plate. It's red because there's red blood cells in there. This is an uninoculated blood auger plate that I incubated just as a negative control, just so we could see the color of the auger here. And again, I know this isn't going to be the best quality. So then, folks, this was a throat swab. I took a sterile cotton swab, and I tried to swab the back of my throat near my tonsils. And it's, I know, again, kind of hard to see, but see that greenish discoloration compared to our uninoculated plate here? Okay, that greenish discoloration, that is called alpha hemolysis. And that's classic for members of your oral microbiome, such as Streptococcus mutans, the guys that form biofilms and plaque on your teeth, and then can cause an increase in dental caries and periodontal disease. And folks, I'm going to take the lid off because there's condensation on it, but you can see there's a variety of different little colonies in there. Our mouths, our throats have more than one type of microbe in there. Some of these little colonies are gamma hemolytic, meaning that there's no hemolysis at all. What I was interested was seeing if there was any beta hemolytic colonies, because that would be suspect either strep pyogenes, or another one that I've actually recovered from my throat is Staphylococcus aureus. So Streptococcus pyogenes is the cause of strep throat, streptococcal pharyngitis. Pyogenes literally means pus producer, so they trigger a really strong inflammatory response. Sometimes you'll see little white dots in the back of your throat where the Streptococcus pyogenes are growing, and that's from the accumulation of white blood cells. And then because Streptococcus pyogenes can trigger this attraction and then killing of the white blood cells as they make toxins called leukocidins, literally white blood cell killers. Because Streptococcus pyogenes is great at making pus. Pyogenes, pyo means pus, and genes means to make. So these are pus-making bacteria. So we call them pyogenic bacteria because they're making the leukocidin. Now, another microbe that I've recovered from the back of my throat is Staphylococcus aureus. And this makes sense because if we're colonized in our nose, it makes sense that we could have Staph aureus that would be moved just from secretions to the back of our throat. So I've also recovered beta hemolytic Staph aureus from the back of my throat. Now, these two little dots that we see here, you might think, oh, that's beta hemolysis, but that's an artifact. I took my swab and I pushed it through the auger. So that's literally light showing through. But indeed, you guys, if we had beta hemolytic Streptococcus pyogenes or Staph aureus, it actually would look just like that. The beta hemolysis is total destruction of the red blood cells. You can read right through it. Okay, so again, the greenish discoloration, folks, that's the alpha hemolysis, probably from Streptococcus mutans. Another pathogen, another member of the genus Streptococcus is Streptococcus pneumonia, which causes pneumonia, and that too would cause the alpha hemolysis, the greenish discoloration. So you guys, most of these plates are going to be really repetitive. This is just another one of my throat where I tried to get some isolated colonies, and again we can see the greenish discoloration, the alpha hemolysis. This was of my gums, and we got lots and lots of alpha hemolysis, that greenish discoloration. Probably I've got lots and lots of Streptococcus mutans on my gums. Probably I should go brush my teeth. Okay, and then this last one, folks, was of my nose. And I know historically, off and on, when I take samples from my nose, I will see sometimes I'm carrying Staphylococcus aureus, the beta hemolytic bacterium. And although it's hard to see here, folks, if I hold this plate up to the light, there's some colonies that look like they might be just starting to carry out beta hemolysis. So I'm going to take these plates home with me, and I'm going to stick them in the garage in a biohazard bag, and I'm going to check them next week and see if maybe we do have some beta hemolytic Staph aureus there. And if so, I'll do another movie so you can see it. All right, folks, so that's that for Blood Agar. And let me see here, the next demo we're going to do is, let's see here, we went from chapter 10, microbial growth media. We went chapter 10, and then I think we did chapter 11, aseptic transfers. So we'll look at our broths, our slants, and see what kind of results we got.